Hi, everyone. Welcome back to our second session on this single topic biliary atresia symposium. In this second session, we are going to talk about research, and we have our top leaders in the field. The title of this second session is Where Do We Come From, and where are we at now, and where do we go from here? With me is Dr. Guillermo Masolini, and I'm Juan Pablo Arab, and I'm going to briefly introduce our speakers. The first speaker will be Dr. Rebecca Wells. Dr. Rebecca Wells is Professor of Medicine and Bioengineering at the University of Pennsylvania, where she is also Vice Chief for Research in Gastroenterology and Hepatology, and co-director of the NSF Center of Engineering and Mechanobiology. Her background is in liver fibrosis. She and her group have a particular interest in the extracellular matrix and mechanics of the liver and bile ducts in injury and repair. So without further ado, Dr. Rebecca Wells, go ahead.
Thank you. Let me share my screen. And can you see this? It's acceptable?
Yes.
Looks
perfect.
okay? Okay, great. Yeah. Thank you very much for the introduction and for the invitation to speak at this conference. I'm truly honored to be part of this group. And what I would like to talk to you today is, in my two talks, is some work that my group has been doing, thinking about biliary atresia from the lens of the fetus, looking through the lens of the fetus. So there is, there are three reasons why we consider the fetus to be at the center of BA and why we've been thinking about the pathogenesis of biliary atresia from the perspective of the fetus. The first is that there's very strong evidence from Dr. Sunny Harpavat, as well as from a number of other investigators, including Dr. Saul Karpin, who's here, that the injury in biliary atresia occurs in the fetal period, that it's prenatal. Second, and I'm going to talk about this more in my second talk, there is compelling evidence that fetal and adult wound healing is very different. And so when we think about the response to injury of in biliary atresia, we need to think about it from the perspective of fetal wound healing. And thirdly, I think it's very important for us to consider that BA progresses markedly after birth, and we need to consider what might actually happen at birth. And I'm going to speculate on that at the very end of my second talk. Sorry, just gotta. I've been asked to give two talks. In the first talk, I'm going to provide some evidence, some published, but much of it not, on susceptibility of the fetal and neonatal bile duct, particularly the extrahepatic bile duct, to injury and fibrosis. And in the second talk, I'm going to talk about the role of matrix in fibrosis progression and postulate that biliary atresia can be viewed as fetal wound healing gone bad. So I'm going to start talking about some data that we published previously. Some of it we published previously in the two publications listed down on the right about the fetal and early neonatal susceptibility to injury. And then I'll talk interspersed through, include some unpublished data. And there are really 5 reasons why we think that the fetal and early neonate may be particularly susceptible to injury. And I've listed them here and also show them in this cartoon. First, if you look at number 1, the fetus and the early neonate have a very patchy cholangiocyte glycocalyx. And I'm going to tell you in a minute more about that and tell you in more detail what the glycocalyx is. The second is that the fetal and neonatal extrahepatic duct has very immature tight junctions, and that it also has decreased cell density compared to the duct, and this is density of the cholangiocyte monolayer, and this leads to increased permeability. Next, if you go to this number 2, the cartoon on the right, there's also an extremely sparse submucosal matrix population in the fetus, and that may lead to increased spread of contents that leak from the lumen of the bile duct into the submucosa. And finally, in the submucosa, there are extremely fibrogenic cells in the fetus and neonate. They're starting to produce the collagen that will provide structure for the duct as the individual moves to adulthood. And these highly fibrogenic cells may be seem to be primed and may also predispose the young duct to fibrosis. I'm also going to talk at the end about the mechanics of the fetal, neonatal, and adult bile duct, and suggest that the response to pressure and changes in that response to pressure might also be very important in susceptibility to injury. I want to first just make a note that the majority of work that I'm going to talk about is done with mouse ducts and primary mouse cells, all taken from the very early neonatal mouse. That's day 0 to day 2 after birth. Now, this is not a fetal mouse, but given that human and fetal extrahepatic bile duct development are out of sync, we think this is actually a reasonable model for the human fetal bile duct. But I just want to bring up that caveat up front. So I first want to talk about the glycocalyx in the fetus and neonate. So the glycocalyx is a protein and carbohydrate polymer layer that's on the surface of a cell, and it's shown to you in cartoon form here. And it has, because it is filled with a number of glycosaminoglycans and very complex, large, and water-attracting carbohydrates, it causes significant excluded volume effects and prevents access in many cases to the cell surface. It's also very highly charged, and Ulrich Boyers's group has proposed that it forms a sort of protective bicarbonate umbrella, and that this bicarbonate umbrella protects against bile acids. In other words, protects cholangiocytes from bile acids in the lumen. And Dr. Boyers has presented very nice data to that effect, and my group has more recently provided data to that effect. So what is the situation with the glycocalyx then in the neonatal extrahepatic duct? Well, first, I want to show you some pictures, some transmission electron microscopy pictures from the adult mouse and the 3-day-old mouse. And if you look at the adult mouse, you'll notice that on the microvilli, on top of the cholangiocyte shown here, it looks fuzzy. Whereas in the neonate, you see very little of that fuzziness. The neonate is on the right. If you look at higher power, and this is shown with the red arrows, you can actually see that there is sort of a fluffy or fuzzy coating on the apical surface of these cholangiocytes. And this is the glycocalyx. During fixation, it actually shrinks, but in many cases, this glycocalyx is as wide as the actual cell itself. If you look at the neonate up close, you see some patchy areas of glycocalyx, but overall, the glycocalyx is significantly diminished compared to that of the adult. And this is similar between the extra and the intrahepatic bile ducts. If we look at it in other ways, in this case, staining with Alcium blue, which picks up some of the mucins that are in the in this glycocalyx, you can see that in the P1 mouse, there is very little Alcium blue staining or mucin staining on the luminal side of the cholangiocyte surface, whereas in the adult, you see a very a prominent sort of teal colored layer in this stain. If we quantify this, and I just want to say all this work was done by Jessica Llewellyn, who's on this, who is attending this conference. You can see that the thickness of this glycocalyx increases progressively as the neonate ages and approaches adulthood. We've also shown this using staining using lectins, which pick up a variety of different carbohydrate epitopes. And we've also looked in the human fetus. We've been able to obtain several samples of human fetal extrahepatic bile duct. As you can imagine, that is very difficult to obtain. And again, we see that there seems to be a lack or a patchiness to the glycocalyx in the fetal extrahepatic bile duct. We have Yu, who's in my lab, has developed a really lovely device that we call a bile duct on a chip. This is really a sort of 4 millimeter long, 160 micron diameter channel that goes through collagen, in which we can culture cholangiocytes in a confluent and highly impermeable monolayer. And so this device has the advantage that we can actually measure permeability very nicely. And we can also apply agents and different toxins, different other injury agents, enzymes, et cetera, to either the apical surface through these large reservoir ports, or to the basal surface through these side ports. So first what we did was we actually treated the cholangiocytes, neonatal cholangiocytes, on the apical surface with neuraminidase, which disrupts much of the glycocalyx. And we tried treating them with the bile acid GCDC. And then we looked at their permeability and asked, do we see a change in permeability after GCDC treatment if we also treat with neuraminidase? And the answer was, in fact, yes, that if we eliminated the glycocalyx in these cells, we saw that there was significant increased sensitivity to GCDC. You can see that in these, in the sort of the brownish columns compared to the more green central columns, or the blue controls, that there was significant increase in permeability in response to GCDC, indicating damage if we removed the glycocalyx. I'm not showing you here, but this lovely bile duct on a chip device also allows us to demonstrate that the basal side of cholangiocytes is much more sensible to injury from bile acids than the apical side, suggesting that protection of the apical surface and also impermeability are very key to protecting cholangiocytes overall from injury. When we look also at neonatal, at primary neonatal cholangiocytes, we see that they have very immature cell-cell junctions. So if we look at, these are cholangiocytes, primary cholangiocytes from either a P3 neonatal mouse, a day of life 3, or an adult mouse. And we've cultured them in 3D to form hollow spheroids. And when we stain those spheroids against ZO1, which is a marker of the cell-cell junctions, we see that it's very irregular and patchy in the neonate and looks much more regular in the adult. We see similar findings if we look using electron microscopy that show that the tight junctions of the neonate are extremely immature and poorly formed. And how does this actually correlate with permeability? Because that is, of course, the key question. So if we take neonatal cholangiocytes, and adult cholangiocytes, and again, allow them to form these hollow spheroids in 3 dimensions. And we load them using rhodamine 1,2,3, wash them and look at leakage of the dye. We see significantly faster leakage from the neonatal cells, those are in red in the graph on the right-hand side, than we do from adult cells, suggesting that neonatal cholangiocytes at baseline are leakier than adult cholangiocytes. We've also looked at neonatal and adult cells in this bile duct on a chip. If you focus, please, on the left-hand side, first we were able to show that the density of the monolayer, of the cholangiocyte monolayer in the bile duct on a chip determined how permeable the cells and how permeable that monolayer was. So if you look at these two middle photos on the left, you can see we have flowed through fluorescently labeled dextran into this channel, and you can see that in these compact, when the cells are compact, and that means they're even more dense than in when they've just become confluent, we see that there is effectively no leakage at all of the dextran of really almost any size, that even going down to 4 kilodalton size from these through these monolayers. We see a little bit more leakage in confluent, but it's very clear that the density of the cholangiocytes in this device and in the monolayer determines in large part how permeable they are. So then when we go over and look at the density of cholangiocytes in a mouse neonatal extrahepatic bile duct compared to an adult, you can see the cholangiocytes in blue here. And you in my lab, who has done all this work with the bile duct on a chip, has been able to show that there is a significant decrease in the density of cholangiocytes in the monolayer of the extrahepatic bile duct of the neonate. And this strongly suggests that although it would be extremely difficult, if not impossible, to actually measure in vivo, that in the neonate, there is a significant increase in permeability compared to the adult in the extrahepatic bile duct. So thus far, I've shown you that the fetal extrahepatic bile duct at the level of the lumen and the mucosa has susceptibility potentially to injury from a patchy glycocalyx on the apical surface of cholangiocytes, that it has immature tight junctions and decreased cell density and increased permeability. I now want to show you that there's a very sparse submucosal barrier function and highly fibrogenic cells in the submucosa, and that this leads to an abnormal response to pressure. So first of all, when we looked by electron microscopy, what we saw in a course of a mouse neonatal bile ducts, we saw almost no collagen in the day of life 1 mouse. And if you look over here at the top on the left, you see a lot of white space and very little of these circular collagen fibers. As the mouse ages, you can see more and more collagen fibers are deposited. And by the time the mouse reaches day 15 and then adulthood, you see very thick collagen bundles. And this is quantified in these 2 graphs here. The fibers are thicker in diameter as the animal ages, and there's also much more significant coverage of the submucosal space as aging occurs. If we look at the neonatal submucosa of a collagen GFP mouse, in which any cell that is actively secreting collagen is green, and I'm very grateful to Tatiana Kiseleva for these mice, we see that in the mouse on the day of birth, there's an extraordinary number of collagen secreting cells in the submucosa, and these are in completely normal mice. And so this is undoubtedly how the mouse goes from having a submucosa with no collagen bundles to the adult where it's just packed with collagen bundles. And you can see by the time adulthood occurs, there are almost no actively collagen secreting cells present. And this is important because it suggests that if you have injury to the submucosa at the time of birth or in the very young animal, there are these cells that are already producing collagen and are therefore very primed to lead to fibrosis. So I've shown you so far that there are multiple points of susceptibility on both the apical surface and junctions of cholangiocytes, as well as in the submucosa, multiple points of susceptibility to injury. The duct is less well protected from injury in the neonate, and that it's primed for a fibrogenic response. So I want to just show you now some unpublished data that Jess Llewellyn generated in collaboration with Amelia Roberts and Rika Soyan, using a technique called pressure myography that's used to measure the response of tubes to increase pressure. And this is a technique that's usually used to study blood vessels, but we've used it, I think, for the first time in the extrahepatic bile duct. And what you do is you take an extrahepatic bile duct, cannulate it on either end, and you fill it and measure the distension in response to increased filling pressure. And that's what this movie here on the right shows you, that as it's being filled, the duct dilates, and how much the duct distends and dilates in response to a specific pressure tells you about the mechanics of that duct. And I will just tell you from work in vessels, the mechanical properties of the duct are determined primarily by the presence of collagen. Sorry, the mechanical properties of whatever is being measured are determined primarily by the presence of collagen and elastin. And we thought it would be very important to look at the neonatal bile duct because, as you can see here in the mouse, there's almost no collagen at birth, as I've shown you before. And similarly, in the lower panel, the blue and the red pictures, there's almost no elastin. Elastin is in the red stain. So there's almost no elastin until adulthood. So it's extraordinarily difficult actually to do this technique, but Jess was able to dissect out ducts from neonatal and adult rats. So we validated that collagen and elastin were the same in the neonatal and adult rat duct as in the mouse, so we actually carried out this procedure using rat. I'll show you mouse data in a minute, but I can tell you that the mouse adult and the rat adult have very, very similar curves. We are able to do this with an adult mouse, but not the neonatal mouse. So if you look at this graph on the left, and if you look at the blue curve, this is the curve of millimeters of mercury of pressure, of filling pressure against the percentage or the increase in stretch. And you can see that this curve really has two parts, this dark blue curve. The first part of the curve actually represents elastin, the contribution of elastin to the mechanics of the duct. And then the second part of the curve, as it becomes more vertical, represents the contributions of collagen. So when we then look at the normal neonatal rat duct, as shown in the light blue, you see that there are differences in both the horizontal elastin component and in the vertical collagen component. And this makes sense because we know from the stains that I just showed you, as well as other stains of rat, that in the neonate, there is very little collagen and elastin. And so you would predict that both parts of these curves would be significantly different. When we use these data to then calculate the stiffness of the duct, we find that at the normal physiological pressure within the duct, and this is derived from a number of different reports in the literature, there's very little difference in the pressure and the stiffness of the neonatal and the adult rat duct. However, if we look at pressures that are higher and are consistent with what people have measured for obstruction in the bile duct of a rodent, we find that the neonate is much softer and the adult is much stiffer at those obstruction pressures. And so this may suggest that the neonatal duct stretches more and is less stiff, and so is potentially protected against obstruction on the basis of its mechanics. But let me just tell you one additional small story as to when this gets interesting, and then I'll finish up. So we became interested when we found these data in a report from 2018 from Nancy Spinner and Marcella Devoto at Children's Hospital of Philadelphia, just right next door to where my lab is. They did a GWAS, actually 2 GWAS studies, looking for susceptibility genes in 2 different cohorts of BA patients. One that seemed to be isolated and one cohort that was syndromic. And in both cases, they identified the locus for a gene called EFEMP1. And although in neither case did they achieve the standard genome-wide significance that you'd see in a GWAS, in both cases, the significance was considered to be suggestive. And this was very intriguing because EFEMP1 is a secreted glycoprotein that goes into the extracellular matrix. And it's part of a large family, the members of which seem to be important in the development of elastic fibers. So although EFEMP1, which is also called fibrillin 3, is not well understood, the EFEMP1 null mouse has decreased elastic fibers in the fascia and connective tissue, although the large elastic fibers in the lungs, for example, in the large vessels, were normal. And that made us wonder what it might look like in the bile duct. So we obtained these mice, and I'm very grateful to Dr. Marmerstein from Mayo Clinic for providing them. We found in our colony the knockouts were not viable. I'm happy to talk to people about that afterwards if they would like to discuss. But we used the heterozygotes, and what we found was that neither collagen nor elastin deposition was changed. It seemed in the heterozygotes to be identical, nearly identical in the wild type and in the het. However, the number of elastic fibers where elastin is organized into fibers was significantly reduced in the EFMP1 heterozygous mouse. And that's these two red images in the middle row, and you can see the quantification. And that made us ask, could there be an impact on duct mechanics as a result of this, this partial knockdown of EFMP1? And so we again, with Amelia Roberts and Rika Soyan, Jess Llewellyn dissected out, and in this case, mouse extrahepatic bile ducts from adult litter mate as well as EFMP1 heterozygous mice. And what they saw was that there was significant difference in the patterns for these two sort of in the stress stretch curves given these two genotypes. So if you look at the middle panel, the collagen parts of the curve on the right are nearly identical, suggesting that that collagen is not contributing to this change. However, there's a significant difference in the elastin component of the curve, and that's the nearly horizontal component, as you might expect, given the staining suggesting that elastic fibers are abnormal. If we then look at what the stiffness of these ducts is at both physiological and obstruction pressures, as I mentioned for the rats, we again see that there's not much difference normally. But if there's obstruction, in this case, interestingly enough, the EFMP1 heterozygote has a stiffer duct than the wild type. And what this suggests, when you have a stiffer duct, it suggests that there may be activation and therefore collagen deposition by fibroblasts that respond to stiffness by becoming fibrogenic. And so overall, this suggested to us that fetal and neonatal duct susceptibility and protection mechanics may be very relevant in BA, although it seems that the neonate is normally protected against damage from obstruction on the basis of these mechanics. Clearly, this is an example of a genetic susceptibility linkage that may actually be relevant to the damage that's seen in BA. So I want to just conclude here by saying that we've shown that the apical protection of cholangiocytes in the neonate is weak, thanks to the glycocalyx and increased permeability. And leakage is likely to spread very easily through the submucosa because there's no collagen barrier. It's mostly hyaluronic acid. The basolateral surface of cholangiocytes is much more susceptible to injury from bile acids. So the fact that cholangiocytes are very permeable and bile acids or whatever else is in bile may spread through the submucosa means that injury may be much more likely to spread. And that in the submucosa, there are cells that are primed to respond in a fibrogenic way. And finally, that mechanics may actually impact the activation and the activation of those cells and the overall response to the myofibroblasts, to obstruction. So I just want to finish by acknowledging the people in my group who did this set of work, in particular, Jess Llewellyn, who's shown at the top here. Gary Kandekar, a former member of the lab, was also important. And they had help from Udu, Kapish Gupta, and Iris DeJong, shown here. And I'm very grateful to all my collaborators, particularly Rika Soyan and Amelia Roberts, for the pressure myography. So I'll finish here and take any questions.
Thank you very much, Dr. Well, for the great talk. Just to remember our audience that you can make your questions on the bottom of the screen. You have the Q&A box, so you can ask any question if you have, and we are going to select and ask to our panelists. And also you have the interpretation button. It's in the bottom right of your screen, and you can select the language. So while we wait for question, I will make the first. So basically, you told us that neonatal susceptibility to biliary injury is at least in part based on the increased permeability. So do you think there is any role of the biliary microbiota on this disease since the permeability is impaired?
I think probably Dr. Becerra could comment on that, but certainly I think that if the microbiota of the bile duct, yes, could have a significant impact in this setting where there is increased permeability in the neonate. So definitely, if there are microbes in the fetal extrahepatic bile duct, they would be more likely than in the adult to be able to transmigrate into the submucosa. Yes.
Thank you. Dr. Karpen also has a question. If the composition of the pressure experiment fluid was similar to the bile?
It was, that's an excellent point. We use, I think it was PBS or something similar. Yeah, some isotonic solution, but we didn't use bile itself. I don't think that would have been possible given this setup, but it's an excellent point. Yeah.
Dr. Becerra, do you have a question also?
Yeah, Becky, a great talk. I'm wondering if you checked if you check a glycocalyx production using organoids or these spheroids from neonatal mice.
We have not checked that in spheroids, but we have looked at, it's actually a slide I took out. We have looked at neonatal mice at all different ages from the time of birth through adulthood and have used lectin staining to characterize, to demonstrate that the glycocalyx is quite different and quite sparse in the neonate to the adult. In the actual spheroid model system, though, we haven't done those studies, but it would be a good idea. I think one thing we have done, however, is I think a better model than our spheroids is the bile duct on a chip, because you have this highly impermeable confluent monolayer with permeability characteristics that are similar to what's been measured in vivo, at least has been published. It's completely tight, and we have been able to show that the glycocalyx forms as we would expect, given our other studies with neonatal and adult cholangiocytes. So in that model system, yes. So I would expect the spheroids would track. Yeah.
Dr. Wells?
Yes.
I'm sorry, considering the susceptibility of fetal liver biliary ducts to injury, do you know if there is a role for aquaporin channels in the biliary duct permeability, in the increased permeability of the biliary duct?
The answer is I don't know. I think that most if not all of what we see can be explained by differences in density and by differences in tight junctions, but we have not specifically looked at aquaporins. Yeah. Colleagues of ours have done a significant amount of computational modeling to look at permeability as a function of density, and our data seem consistent with what they find just purely based on density. But we haven't looked at aquaporins.
Thank you.
And I have a last question here from the public. So understanding that the matrix of collagen, periductal collagen, is much lower than in neonatal states. However, when they are activated by fibrogenic factors, is this increasing quickly?
So we are actually working, we are in the process. We've developed methods to isolate the fibroblasts of the neonatal submucosa. As you can imagine, that is easier said than done. We do know that they have different mechanical responses to stiffness than adult fibroblasts, submucosal fibroblasts do, but we haven't yet gone through and tested in detail all the different profibrogenic factors, but that is definitely something that we're doing.
Thank you.
Please, another question, last question, please. Juan Pablo, from Gabriel Melgarejo, pediatric surgeon from Colombia. According to your conclusions, what would be the next steps in these investigations? And could you please give us a possible medical applications for the everyday practice, please?
That is the key question, yes, to this and the next talk. So I think that I'm not involved in the care of biliary atresia patients, and so I can't comment on the use of URSO or other agents. But I do think it's very intriguing that our data suggests that in the injured duct, leakage of bile into the submucosa and leakage of, the particularly hydrophobic bile acids in particular might be especially damaging. And so the possibility that you could actually switch the composition of bile to less damaging bile acids, and there is the thought that that could lead to less submucosal and therefore mucosal damage. So that's one potential application of this work. Yeah.
Thanks.
Thank you. I know everyone has questions, but save some for the next talk, and then we can continue. We are on time. So Dr. Masolini will introduce our next speaker.